Today's presentation will be on curing scurvy. My name is Jay Farah.
I'm Connor.
I'm Simra.
Going to the first slide, looking at the background information and justification. Many species such as teleost fishes, anthropoid primates, guinea pigs, and some birds all lack the ability to biosynthesize vitamin C. Humans and orangutans both contain the non-functioning gene, also known as the pseudogene, that codes for the enzyme galonolactone oxidase, which is responsible for the formation of vitamin C. The gene is non-functional because of a mutation that has resulted in the loss of 7 out of the 12 exons. Addressing this problem could potentially cure the disease of scurvy that occurs in orangutans when they do not get enough vitamin C. Vitamin C is responsible for growth, development, and repair of body tissue. A lack of vitamin C can potentially lead to a disease called scurvy, which leads to symptoms such as bruising, excessive bleeding, weakness, fatigue, rashes, and other symptoms. The results from the genetic modification in orangutans can be used for future application to help further find a cure for scurvy in humans. The use of CRISPR to transfer the gulo gene is an appropriate solution to this problem because it's the only way to implant the gene into the genetic sequence of an orangutan. Now, looking forward at the worldwide impacts of scurvy, approximately 13% of the US population is vitamin C deficient. And a large majority of the populations that encompass this statistic do happen to come from low-income families. And since 2009 and up till 2014, the number of people in England affected by scurvy has increased by 27%, which further emphasizes the importance of this issue. And in third world countries, vitamin C deficiency has had an even larger impact, as malnutrition is a big problem in Africa. And in northern India, it's up to 73.9%. And looking at the experimental plan, the idea is that the gene that we have chosen to work with is galonolactone L- oxidase, also known as GULO. This gene was chosen as it is responsible for the production of vitamin C. The molecular function of this gene is to biosynthesize vitamin C from a substrate known as L-galunolactone. This gene does not have any effect on the phenotype. We have then chosen to use 15 Sumatran orangutans and 1 pigeon as our organisms of choice. We choose orangutans because they are one of the primates that possess the non-functioning gulo gene, and they're similar, genetically speaking, to humans. In order to complete our research on orangutans, we are not buying them from a supplier. However, we're going to collaborate with an orangutan sanctuary to carry out the project and ensure that all ethical procedures are conducted in order to make sure that the orangutans are treated ethically. We plan on using CRISPR to insert a functioning gulo gene from another pigeon that we'll gather into an orangutan. And once this gene is inserted, we hope that the orangutan will be able to biosynthesize vitamin C, since the functioning gulo gene in the pigeon will be transferred into the genetic sequence of the orangutans. And so on to the next slide, we see the feasibility and innovation and the practicality of the project. The project is quite feasible since genetic insertion using CRISPR has been tested many times and has succeeded in the past. And we've already located a functioning gulo gene, which we're going to be retracting from a pigeon. And the reason it's innovative is that it's never actually been done before, where they have managed to basically cure scurvy using CRISPR. So, and basically humans would be able to then biosynthesize vitamin C, eliminating the existence of scurvy in the future.
So the first part of this experiment will be gene isolation. mRNA will be isolated from the liver tissue of a pigeon and will be used to synthesize cDNA using reverse transcriptase. From this cDNA strand, a double-stranded cDNA will be made using DNA polymerase, and then PCR will be used to isolate the gulo gene. Figure 4 shows a visual of how forward and reverse primers are used to amplify the gene along with the help of the Taq polymerase. So the next part for this experiment is creating recombinant plasmids. With PEF-GFP vectors, a SMA1 restriction enzyme will cut right after the EF1 alpha promoter, and the BSU361 restriction enzyme will cut directly after the gene that is already present in the vector. Next, the isolated gulo gene will be put into the plasmid at the cleaved ends using DNA ligase. The plasmids will then be introduced to E. coli and treated with ampicillin. Since the plasmids being used contain an ampicillin resistance gene, only cells containing the plasmid will be isolated, and then the plasmids will then be isolated from the E. coli cells using lysis and denaturation. The last part of this experiment is the integration of the gene into cells in the host chromosome. The vectors will be injected into the liver of the orangutans where vitamin C is biosynthesized. Once injected, transfection and CRISPR-Cas9 can take place. So for transfection, the lipids designed for plasmid transportation first envelope the plasmid. The cations and the lipid then bind to the negatively charged nucleic acids of the plasmid. And once the plasmids are introduced into the organism, the lipids will fuse with the phospholipid bilayer of the eukaryotic cell and release the vector into the host cell where it can be transported to the nucleus. For CRISPR-Cas9, the CRISPR protein complex will also be added to the host cell where it will cut the genome on chromosome 8 along with gRNA. Although it may not happen every time, the goal is that the vector will insert itself where the cut was made by CRISPR-Cas9. This will then integrate the gene into the host genome. In order to determine if the objective was met, blood assessments will be performed before and after the gene is integrated into the host to determine if the vitamin C levels have increased and if the gene that was inserted is functional.
All right, here is our vector diagram where you can see the gulo gene as well as the EF1 alpha promoter that drives its expression. The SMAL and BSU361 restriction sites are also shown, in addition to the ampicillin resistance gene, which allows us to isolate the cells containing the plasmid. And once the vector is integrated within the host, the liver cells will express the gene. Here is our phylogenetic tree. The tree shows the evolutionary history of the gulo gene. The letters represent the exons of the gene, and as you advance up the tree, it is less likely that the organism is to have the functioning gene present. You can also see how the exons change over time. The gene loses functioning for most organisms above the horizontal line that is placed above birds. Most mammals have non-functioning pseudogenes as seen with the dog, cat, and panda. Other mammals, such as some bats, rodents, and primates, have lost critical exons due to mutations, including nonsense mutations, which shorten or interrupt exons. Some mammals have pseudogenes that can occasionally biosynthesize, but they are losing this ability. This is how we see the loss of the functioning gene in orangutans. Some rodents and bats can have the functioning gulo genes, such as rats and a species of fruit bat known as Rosetus leschinalti. These species are more closely related to our target organism than our pigeons, and they could potentially be better source organisms, but studies show that the abilities of these organisms to biosynthesize vitamin C are on the decline. Birds are the most closely related group of organisms that have a functioning gulo gene. After that, some organisms, as explained before, have the functioning gene, but these genes are not perfect replicas. And the significance of us using birds is that the distant relative is it is likely that there will be a successful exchange of genetic information, is not too likely, but it is not impossible, which is the important part. If this were to work on orangutans, the implications would be great. Primates have very similar genetics, and success for orangutans could very well mean the success for humans and other primates. Humans and orangutans share 97% of genes. Humans also have similar mutations as orangutans, resulting in the non-function of the gulo gene. The impact of creating these GMOs are uncertain. If successful, the organism would have an advantageous trait over competitors, but these species have thrived without this gene for a very long time, so it is likely evolution would take place, but over a very long period of time that would be essentially unobservable. Ethically, this experiment has been completed on mice before, and it was successful. no mice were harmed. So since we are conducting the same experiment but on a different model organism, we anticipate that none of the orangutans will be harmed. Since this experiment has not been done on larger mammals, we chose to use orangutans as model organisms before testing on humans. During the duration of this experiment, orangutans will continue to live at the sanctuary and continue to be cared for by the sanctuary facilities. If the experiment is successful, we intend to apply the same concept to humans. Because CRISPR technology is not completely accurate, the liver of the orangutans may be damaged. In order to prevent health issues that could occur in orangutans or pigeons, the health and survivability of the animals will be prioritized. Moreover, the use of the Guide for the Care and Use of Laboratory Animals from the National Research Council of the National Academic Council will be followed. For future directions, after confirming success of biosynthesis in orangutans, trials with other primates such as baboons or simians could commence in a similar fashion. The presence of the gulo gene in all primates allows for an identical process of transfection and integration through CRISPR. Turn the gene on after creating cDNA to isolate the working gene from the pigeon. The ultimate goal would be to perform trials on humans and make gene therapy available in order to reduce the number of people affected by scurvy. An alternative direction could include curing scurvy in a number of different mammals, such as the guinea pig, through the same methods in order to increase the longevity of their lives. We could also take a step back and assess the levels of success when using other general mammalian expression promoters, such as the CMV or SV40 promoters, and compare them with the EF1 alpha promoter used for this project to determine which might be more efficient in expressing the gulo gene. That was our presentation on curing scurvy, and we hope you enjoyed.